Hello everyone, welcome to the Module 2 instructor presentation. This presentation will cover some of the concepts found in Chapter 3 and 4 of the text and other material found in Module 2. Here you can see some of the more important concepts and themes that I want to go over today. This all of course has to do with the visual system, but one of the things you'll find is if you understand some of these concepts that pertain to the visual system, they help you understand the brain in general. We have parallel processing, retinotopic organization, how receptive fields are organized, feature detectors, and hypercolumns, and this concept of modularity. Uh, and all of these things will come back throughout the course. So understanding them now helps you as you progress through the course. One of the concepts that can be tricky for students is the concept of the receptive field. So you've already, in your readings and in your activities, you've learned about the retina, you've learned about ganglion cells, you've learned about some of the parts of the brain. Receptive fields, when we're talking about the visual system, these are properties of neurons. And here you can see the textbook definition, area of receptors that affects firing rate of a given neuron in the circuit. The key word here is effects. What will happen is cells, let's say we're talking about here, we do this, by the way, by recording the activity of a particular neuron, which can be done using an electrode. But sometimes you can present a visual stimulus to an animal and it might make certain neurons more active, and in other cases, it can make it less active. And receptive fields of various parts of the visual system can vary in terms of different types of characteristics. Some neuronal receptive fields are big, some are smaller, some tend to be driven more by movement, some more by, let's say, we might say color and other things. So the receptive field properties are something that neurophysiologists have been studying for a long time. And one of the things we know is if we're talking about single fibers in the optic nerve, this refers to ganglion cells, is that you have, the cell can either increase its firing or decrease its firing. That's basically all neurons do. But what's kind of interesting is that in here you can see the general organization of a cell of a ganglion cell. And by the way, these are also this type of organization, which is called center surround. See how they're circular? Think of this as a little tiny patch, a little spot in the visual field. All right. So everywhere you look, there's the visual field. As you move your eyes and move your head, that visual field is always in front of you, and you've got parts of your retina, specific as objects move about in your retina, they are stimulating different parts of that. They're stimulating different ganglion cells that serve different portions of that retinal of that visual field. And some of them have this center surround organization where, let's say, if this spot of light where my mouse is, it happens to go here in the center part, it'll make the cell fire more. And then if it happens to wander out here, it's going to make the cell fire less. So this cell can actually discriminate. It's showing it will actually vary its firing depending on where the spot of light will be located. And it turns out that ganglion cells behave this way. They have this center surround organization. We also see this in cells of the thalamus. And here we see that it's also centered around, but here if my spot of light were to go in the middle, it actually makes the cell fire less, and the outside, the surround, it has the opposite. And so you can see here, this is all in your book, by the way. Once again, make sure you look at your book. Um, Here you can see if I put a spot of light right here in the middle, but not completely filling up that center part, the cell's firing here pretty good. But then if I completely fill it up, you can see it's a great deal of firing in the cell. But as I start to put the stimulus in the surround region, you get inhibition. All right. So this is how cells at the ganglion level in the retina work. This is also how cells in the lateral geniculate nucleus of the of the thalamus work, the LGN. And so here you can see once again that's the summary. Um, we call this center surround antagonism. So the center of that receptive field, this circular receptive field, is different than its surrounding region. All right, if that makes sense. Um, by the way, here you see Hubel and Wiesel. These are the gentlemen that really did a lot of pioneering work in receptive field properties of neurons in the visual system. They looked at, in particular, the cortex, which we'll get to in a second here. But one of the things that's interesting is that here's that visual field, here's a cat, and incidentally, a lot of this work was done with cats. But here you can see, imagine this is like a spot of light being moved from this part of the visual field to the center, downward in the lower part of the visual field. Here you can see that as it moves from going from A to B to C, you're stimulating different parts of the retina. So here's A. Remember that image is inverted. That's why A is down here. Then you got your B, then you got your C moving about. This, by the way, this is also a concept that's important. This is retinotopic mapping, where there are, out here in the visual field, there are specific parts of the retina that get stimulated. All right, and they're showing that here. So one of the important things to figure out with receptive field is where in the, here, the cells at A, the visual field that's important is located at point A, B and point B, C and point C, and so forth. And as I said, LGN also have the center surround receptive fields. And from the lateral geniculate nucleus, signals are sent to the back of the brain, called the visual cortex in the occipital lobe. There's plenty of pictures, by the way, in your text, so be sure to look at that. So you start off at the retina, and here's here's a little bit of an image just from your text. Here you start off at the eyeball. It ends up, here's the thalamus, and then from the thalamus, this is the signal, right? So then the signal goes from the thalamus to the back of the head. This is the occipital lobe. And in the very, very back, where my mouse pointer is called V1, or the striate cortex. Striate, by the way, means layered, because there's 6 layers of the cortex. But there's a lot of different names for this, but we'll just call it visual area 1, or V1. And so this is the primary visual pathway. And here you can see, kind of looking at it from the bottom there, here's your eyes. You have these connections going from the eye out to the thalamus, and then back here to the visual cortex. One thing to note, while I'm thinking about it here, is that essentially what happens is the right, let's say if we're talking about, if this, kind of looking up here, this would be, I guess, is that the right or the left eye? I guess that's the left. Here you can see this particular eyeball, the visual field here, which will be on this half where my pointer is processed by the other side, by the other hemisphere. This is called contralateral processing, contra meaning other. So your right visual field is being processed by your left brain, and your left visual field is being processed by the right brain. Incidentally, just to let you know, now they're connected because there's fibers that connect both hemispheres, but that's just how the wiring is, just to let you know. Now, when we get to the visual cortex, what Hubel and Wiesel did their work on, this was done years ago, back in like the 1950s, 1960s, a long time ago. They basically laid the groundwork for what we call feature detectors. All right, they found that once that signal reaches the cortex, that the receptive fields, they would locate receptive fields of neurons in the animal, but you couldn't just have a spot of light there, or something like that. You had to have something more specific. You had to have maybe a line or an edge, and maybe the stimuli had to move, or this, that, and the other. So the receptive field properties became more and more specific, but they're still pretty broad. We're talking about just like edges and things of that nature. And they identified different types of cells, simple cells, complex cells, and what they called end-stopped cells. And this is it. Pathway from the retina shows neurons that fire to more complex stimuli. And you'll, of course, see in your text that as we get even further along in the visual system, you have cells that respond to maybe more specifically to parts of the body, or maybe the face, which is quite important for our visual system. And here's what they found is that the receptive fields, they actually change their structure, no longer center surround, but maybe this one is what we call a side-by-side. So if a line, a lot of the receptive fields of visual cortical neurons tend to respond to, let's say, a line. And as I get that line and I sweep it through here, going back and forth, if I'm in the middle of that receptive field for this particular cell, the cell's going to fire more. Things like that. And that's what's shown here. Here's side by side. This would be the surround, and here is the center. If I put that bar in the center, I'm getting elevated activities. But if I go, let's say I turn that, if I change the angle of that line, I get less firing. But we also find that you're going to have cells that might respond to this, maybe more like a 45-degree angle. So there's a whole range of orientation sensitivity here, and that's what's shown in your text. So that particular cell It's going to show the most firing when the stimulus is a vertical line, and it's going to go less and less as I turn it to the side. And they actually found this sort of accidentally. Back then, in order to get a visual stimulus, they didn't have computers software to do any of that kind of stuff like you would do it now with more advanced screens. Back then, they just had a projector, and when you put these projector slides through there, there's a little, like a line, that's like a place of contrast between the slide and the background. And they accidentally found, while they were recording cells in the cat, that as they moved the slide up and down, they found that, wow, they got the particular cell they were recording to fire more. See, what they were initially doing was putting like circles and stuff. And Cortex doesn't, what we find in the Cortex is it tends to respond more to edges, lines, edges, maybe like a corner here where my mouse is. Like it might, you might have some cells that respond to a corner of a particular orientation, all kinds of stuff. Once again, these are all feature detectors. And here you can see a summary of the properties of these cells, going from your ganglion cell all the way up to these three types here: the simple cortical, complex, and end stopped. These are all feature detectors, and you can see they're the types of things they respond to. So the idea is that as we get further in the system, you got your retina, the ganglion cells of the retina sending signals to the lateral geniculate, and then the lateral geniculate, and the ganglion, these are just basically responding to spots of light. They're more generalized in function. They send their input to these feature detectors in the cortex, and now we're seeing a bit more specificity. Bars, maybe the bar has to move, or the bar has to be a certain orientation, things like that. So we see that. So this is the essence of what we call a bottom-up perspective, that we're kind of, as we get further in the system, layer upon layer of visual information is being added. And I already mentioned this. We have a map of the retina on the cortex. So this is very interesting. We have an actual map of the visual field onto the retina. Just think of it as a copy of the outside world visually that the cortex have, and this map is maintained throughout the visual system. All right, so this helps us, of course, be able to, because of this mapping, we can precisely locate things in our visual field. It allows us, if someone throws a ball at you, to precisely know exactly where it is and be able to catch it, as an example. The mapping is kind of interesting. It's not it is distorted in the sense that foveal region of your retina, right here, this is the fovea, right here in the central part, more like where B is. This is your central vision, and when going through chapter 2, you've learned that we have the rod vision, we have the cone vision, and in this area here, cone vision tends to get proportionally more cortical space allotted to it than the peripheral retina, and so a really tiny portion of the cortex, a tiny portion of your retina, this little part here, gets proportionally far more of the visual cortex to process it. This is called cortical magnification factor. So you can see here the fovea only accounts for 0.01% of the retina, but yet it's given almost 10% of the cortical maps area. This explains why we can do such, we can use our visual system for, fine detail, to really examine objects, look at the fine details of the structure of objects. Out here in the periphery, this is more of a shadow detection, motion detection, and, we're not using this so much for identifying what things are, but more about where things are. So this would be more of your what versus where. We have to foveate to be able to know what things are. Okay, let's see. Now another concept that I want you to understand that can be difficult is this notion of the columns in the cortex. All right, so the visual cortex, what Hubel and Wiesel found was if they put that electrode, as they recorded it, if they put it down in a particular area going vertically down through the top of the brain down to the bottom, that they had receptive fields that were more or less in the same location within a particular column of cells. And what they found was, here you can see, so imagine here's a schematic. This would be the surface of the brain. They put the electrode down there, and this would be neurons. This would be a layer 1, layer 2, layer 3, layer 4. More, it's not perfect, but more or less they found overlapping visual fields. Receptive fields that. So the idea is that there are, cells 1, 2, 3, and 4 are more or less processing the same part of that visual field, and we call that a location column. All right. And they also found evidence for what we call orientation columns. Neurons within columns fire maximally to the same orientation, like vertical versus horizontal. Um, this is the concept, though, that I want you to understand, that about a millimeter of cortex, millimeter cubed, basically, it's going to be a little processing module for a part of the visual field. Okay, and so also there's ocular dominance. Some of these cells preferentially are going to respond to either the right or the left eye in those columns. And here you can see again, like you might have here, putting that electrode down there. All of this particular column that they found, that they have, all the cells tend to respond to vertical lines, versus this particular group of cells is responding to vertical. This is horizontal versus vertical, for example. But if I go through all what they found, this is showing you a hypercolumn basically in one particular location in the, or not, it's not, more or less a particular location in the visual field. You have cells. You have a group of this hypercolumn. You have a group of these smaller columns. These are all different location columns that respond to vertical lines. They're not showing in this picture, but you also have within this column the whole range of orientation selectivity. So you might have another group of cells that are going to respond to the, let's say, horizontal, or maybe cells that respond to 45-degree angles and such. And this would represent a hypercolumn. And the theory is that all of these columns work together to cover the entire visual field. Is a concept your author goes into. It's called tiling. So here you can see this picture. This is very schematic. It's not done. This is a little bit of a simplification, but the idea is that as you get all of these different hypercolumns, you'll have one particular hypercolumn that's maybe processing this little part of the visual scene. Then a neighboring one is processing this part of the scene. And then here's another one, a neighboring one processing this part of the scene. So all of these hypercolumns collectively allow us to at least initially process this visual scene, this forest or whatever, the trees that we're looking at there. Now there are a lot more hypercolumns than what's shown here. I think there's about 180,000 of them in the cortex. So this is an oversimplification to kind of just so that you understand the concept. But the hypercolumn is evidence that we have distinct processing modules in the visual system, all working in parallel together, so that we can, instantly perceive this scene. Okay, I know I've just thrown a lot of information at you, so feel free to go back to this and also keep look at this in the text. One of the final concepts I wanted to address is this notion of the what and where systems. And you can see that, so once we've gone from, this is showing you a macaque brain, by the way, as the information goes back here to the occipital lobe, but then the visual pathway splits off when we get some systems heading more dorsally or to the top, and another stream that we call that's heading ventrally. We start to see more and more evidence for processing in the visual system, parallel processing, modularity, this notion that different parts of the visual system each have their own functions. It turns out that information, or these neural signals that tend to go down here to the bottom part of the temporal lobe, tend to process information about the identification of objects. So make sure you look at that in your text. There's a lot of information about that, and how Ungerleider and Mishkin, who are the pioneering researchers in this area, how they identified that using monkeys and different types of behavioral neuroscience tasks. On the top, going dorsally, this is a pathway that's more involved with locating objects. Okay. Also, we also know now it's also a how pathway. How do we interact with objects up here? So it's, we need to know where things are. And we know how, we need to be aware of how, using our visual sense and the other systems too, but how can we interact with objects? And it turns out that we know that when people have damage to this dorsal pathway, sometimes they have a hard time being able to do a task like, let's say, what we do all the time. We get let's say a credit card, and we have to insert it in a card reader, let's say at Target or wherever you're shopping at. You have to be able to locate the card reader. You've got to be able to locate it, and you've got to orient that card just right so that you can put it in the slot. Folks that have damage up here to this house system, which is in the dorsal part of the brain, tend to have a hard time doing that. They can tell you what it is. This system is fine, so they can say, oh yeah, it's a card reader, no problem. But then it's almost like they almost have a motor problem, which is very interesting. Okay, so that was some of the major concepts found in chapters 3 and 4, modularity, receptive field organization, hypercolumns, and so forth. Make sure you spend some time on that because it can be a little daunting when you first are exposed to these. And make sure you do all the different activities found in the course module. And have a great day.